welcome back to Jivashastra Abhyasa. I'm your educator Narendra Nagde, and I hope you all people are doing good at home in this lockdown, and you are learning and you are keeping up with your learning process. So today we are going to learn about the mechanism of DNA replication, the process of replication, the part 2, where we are going to only learn the mechanism. So let's finish off the first thing. If you are new to this channel, please don't forget to subscribe to the channel because it's easy and free. In the last video, I have asked you a question that was which you didn't answer, and it was, what is the official currency of Japan? And the answer is, the official currency of Japan is Japanese yen. The Japanese yen, and it is symbolized like this. It is symbolized like this, and it is introduced in 1871. If you need more, you can Google it. And today's question is, what is the official currency of United Kingdom? I'll repeat it again. What is the official currency of United Kingdom? If you know the answer, please comment below, and if you don't, so I will answer in my next video. So what's the wait? Let's click. The mechanism of DNA replication includes 5 different major steps, and this particular DNA replication is also known as semi-conservative type of replication. So you have to wait until the end of the video to know what is semi-conservative type of replication. So in 1963, a scientist named John Carin confirms that there is a origin of replication site is there in the E. coli. So he confirms that there is only single origin of replication site is present in the E. coli site. He confirms, and also he confirms for many of the prokaryotes. And in the coming few years, eukaryotes were termed as it is having a multiple or more number of origin of site for the replication. And he also confirms that in 1963 itself he confirms another thing, that is the replication of DNA might be unidirectional or bidirectional. So that means it may from 5' to 3' end only, or it may go for 5' to 3' end and also 3' to 5' end. So this was the John Carin, the scientist, who confirms this theory. And now coming to the mechanism, the first mechanism, or the first step of the mechanism, is activation of steps of mechanism of DNA replication. And the first step is mainly this is a very important step, the activation of deoxyribonucleotides. So as I have mentioned in the last video itself, what are the 4 different kinds of nucleotides? So if you haven't seen that video, please click over here and you will definitely view the last video. So there are 4 different types of deoxyribonucleotides are there, and those are deoxyribonucleotide adenosine, adenine, guanine, cytosine, and thymine. So it is also mainly it is known in the form of deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, and deoxycytidine monophosphate. These will be inactivated. These are present in the nucleosome in the inactivated state. What is that state? It is in inactivated state. This has to get activated. The process of activation of nucleotides are known as, or is known as, phosphorylation. phosphorylation, and this phosphorylation is carried out with the help of ATP. The ATP is used, the energy is used for the process of phosphorylation, and this is catalyzed by an enzyme known as phosphorylase. Phosphorylase. So this is how the activated, or how the particular deoxyribonucleotides are activated, which are known as nucleotide pool inside the nucleosome, which are inactivated, and they will get activated by the process known as phosphorylation with the help of ATP. The activated nucleotides are known as, or the phosphorylated nucleotides are known as ATP, GTP, CTP, and TTP. Okay, these are the activated nucleotides which are now ready to get binded to or which are ready to for the replication and acts as a raw material. Now the second step is unwinding of DNA helix. The second step is unwinding of DNA helix. As you all know that the DNA is a helical structure, or which is made up of it is a DNA helix or double stranded DNA helix. We have studied that, so it somewhat looks like that, not exactly. Here the two important enzymes are going to act to unwind it. One is, you know that, one helicase, and the second one is topoisomerase. And you already know the functions of these two structures. The helicase, it unwinds, and this breaks and reseal the, and this is going to reseal the DNA helix structure. So how they are going to act simultaneously to produce a structure like this. Structure like this. So this part is unwinded. Like if you pull a rope, so which is going to expand. So like this, if you pull a rope, so it is going to open up, right? So but when you leave that rope, it is going to wind up, right? So like that itself, this particular DNA is going to happen. So now these two are pulling it up, and they are making a opening, for replication. And if you leave that, if the action of these two enzymes are over, then it again going to wind up. So that's why a protein is released here, so which is acted upon this particular part. A protein is acted, and that protein is known as helix destabilizing protein. So this protein, so it stabilizes this particular thing. That means it holds the structure like this itself. So that is how these three important enzymes, or these two enzymes and this protein, nothing but the same, they are very important for the formation of the replication bubble. Or also which is known as replication fork, which is Y-shaped replication fork, which exactly looks like this. The next step of the mechanism is formation of RNA primer. Formation of RNA primer. Let it be here itself. So this is the fork. So what I told you before. But the third step, we will study about that later. And the third step is the formation of RNA primer. Formation of RNA primer. So as you will all know that the RNA primer is synthesized by the process or the enzyme known as RNA primase. So it because it is the initiation factor for the replication of DNA. So the RNA primer is the initiating factor, right? Yes, okay, fine. So let's know something about it. So this RNA primer is of 8 to 10 nucleotides. It is having 8 to 10 nucleotides, and it starts with 5' end, 5' phosphate end, 5' phosphate end, and ends with 3' OH end. Okay, so why it is called as a initiating factor? Because it has the ability to bind the nucleotides to the daughter strands. Okay, that's why. Or it has the ability to start the initiation, its ability to start the process of binding. So that's why it is known as primer. RNA primer is known as initiation factor for the process of replication. Now the very important process, that is the fourth process is known as binding, or it is known as base pairing. Base pairing. Okay, the base pairing is the simplest thing. As you all know that all the nucleotides are complementary to each other. That means the binding takes place in the way like this. If ATP is there, so there will be definitely, it will attract to the and there will be binding of thymidine. So if it is a CTP, there will be G is guanosine is going to come, and G only that will be binded to the particular daughter strand. And if there is a GTP, here it is A is going to come, right? And it is TTP, then sorry, here not A, here C, and TTP, A is going to come. So now whenever this particular binding takes place, there is a release of, so here adenosine triphosphate is there, and the adenosine triphosphate will be converted to monophosphate. Okay, monophosphate. So that means this will happen deoxythymidine monophosphate, plus there will be release of 2p. There will be release of 2-phosphate, which and plus energy. Energy is going to be released, and this 2-phosphate which is released from the ATP or the triphosphate, which is converted into monophosphate, it is going to be used in the formation of hydrogen bonds between the two nucleotides. So it is used in the formation of hydrogen bonds between the two nucleotides, and this is how the base pairing takes place. And this base pairing is catalyzed by the enzyme known as DNA polymerase 1. So the enzyme which catalyzes the process is known as DNA polymerase 1. So it increases the activity or the process. Okay, process. And here this complete process is catalyzed by the DNA polymerase 1. But one thing very important, so here you can see the 2-phosphate energy and the binding process, right? So this is catalyzed by another enzyme known as pyrophosphate. This is catalyzed by the pyrophosphate. So this is about the base pairing.
Final stage of the DNA replication, or the mechanism of DNA replication, is initiation and elongation of strand. Initiation and elongation of nucleotide or polynucleotide strand, okay, or DNA strand, whatever you can call it. So now here it comes to the explanation of the fork, that is Y-shaped DNA replication fork. So I'll explain here itself. So it is a very easiest method to explain. So as you all know that DNA polynucleotides strand runs antiparallelly from 5' to 3' and 3' to 5' strand, and now it has been opened and it has formed a place for the replication process. Here the RNA primer is very important, because the RNA primer, it initiates the process of DNA replication. First, it acts on the strand which is 5' to 3' end. So RNA primer is going to act here, over here, for example over here or over here. So it is going to act mainly here. It acts exactly if I tell you. So here it acts, and the process is starts, and this strand here, the nucleotide binding is continuous. There is no gap, and that's why it is known as leading strand, where the process is catalyzed by DNA polymerase 3. That's why this particular strand is continuous, because of the DNA polymerase 3, which catalyzes or increases the rate of reaction, as that of the second strand, which runs from 3' to 5' end. So it doesn't, here there is no action of DNA polymerase 3 takes place. That's why here the Okazaki fragments are created. The Okazaki fragments are the small short, here you can see the Okazaki fragments, the gap, and there is a gap between each Okazaki fragments. The Okazaki fragments, it is found by the named scientist itself, Okazaki. Okay, he's a Japanese. The Okazaki fragments are of 30,000 to 40,000 nucleotides, which has 30,000 to 40,000 nucleotides, and for each time, it's for the each and every formation of Okazaki fragments. So RNA primer is required. So you can see RNA primer is required for the process, for the synthesis of Okazaki fragments, and the Okazaki fragments leave a certain amount of gap, which is filled by, so you can see here gaps are there, gaps. So these gaps are filled by DNA ligase, which binds the sugar and phosphate molecules together or sticks them together, and the strand is completely filled. So this RNA primer will be removed while the binding of nucleotide. Here also, if there is RNA primer, that will be removed and the nucleotide will be binded. So that's why this the strand which runs from 3' to 5' end is known as lagging strand, because it lags in that. There is no continuity. So that's why it is also known as semi-discontinuous strand. It's also known as semi-discontinuous strand. So for all these reasons, so it includes the continuous leading strand, and also it has a lagging strand. That's why this replication of DNA is known as semi-conservative type of replication. So the two strands which are formed on the parent strands are known as daughter strands, and they are going to be released after the formation, and they are going to go into the nucleosome, and the process of cell division takes place after the replication process. So this is what the replication or the mechanism of replication of DNA is. So I hope you like this video, because it is a lengthy video, I know that. So that's why I want to tell you people. So if you like this video, please share, subscribe and like, and hit the like button. So don't follow to subscribe, and it's easy and free. So until next time, let's hit the road of success. Jai Hind.